This overview covers the cell theory and prokaryotic cells. Most cells do not have the round, smooth shape that is usually depicted. Cells are very dynamic, and they come in lots of different shapes and sizes. Cells were discovered in 1665 by Robert Hooke, and his work was ignored until studies of cells were conducted by Schleiden and Schwann in the late 1830s. It was Schleiden and Schwann who proposed the cell theory. The cell theory states, all living things are made of cells. Cells are the smallest living units, and all cells come from pre-existing cells. Theories are based on how the world works today, and today you cannot get a cell from anything other than a pre-existing cell. The cell theory also includes the facts that cells vary in size and shape, and that all cells today represent a continuous line of descent from the first living cells. Even though cells come in different sizes and shapes, surface area to volume ratio is a limiting factor for cell size. This is illustrated here. Cube number 1 is 1 centimeter wide, 1 centimeter tall, and 1 centimeter deep. To calculate the surface area for cube number 1, we multiply length times height. This is the surface area for one side. We multiply this by 6 since there are 6 sides in a cube. Volume is calculated by multiplying length times width times height. The surface area to volume ratio for cube number 1 is 6. The ratio for cube number 2 is 3, and the ratio for cube number 4 is 1.5. You can see that the smallest cube has the largest surface area to volume ratio. Larger surface area to volume ratios allow for a faster rate of material transport into and out of a cell. The faster you can move things in and out, the faster chemical reactions can happen in the cell. Reactions that support life processes have to happen very, very quickly. Thus, the surface area to volume ratio is very important in cells. There are some cell structures that all cells have in common. All cells have genetic material in the form of DNA. All cells have cytoplasm, which is a semi-fluid, gel-like substance that fills the interior space of the cell. And all cells are surrounded by a cell membrane that we call the plasma membrane. I usually abbreviate membrane as MBN. The plasma membrane is basically a phospholipid bilayer, and we learned that a bilayer is one of the shapes that phospholipids self-assemble into when we covered this subcategory of lipids. The last structure that all cells have in common are ribosomes. When we covered nucleic acids, we learned that ribosomes function in making proteins. Ribosomes are composed of ribosomal RNA and proteins. There are two basic types of cells, prokaryotic cells and eukaryotic cells. Prokaryotic cells have a very simple internal structure, and their most distinguishing feature is that they do not have a membrane-bound nucleus containing DNA. The DNA is not isolated from the rest of the cell. Prokaryotic cells are found in the domains bacteria and archaea. They are generally single-celled organisms. They're found individually or they can be found in strings of cells or clusters that live together, and they are adapted to most types of environments. You find them everywhere. Typical structures that you find in a prokaryotic cell include DNA in the form of a single circular molecule all scrunched up like spaghetti, and it's sitting in an area of the cytoplasm known as the nucleoid region. There is no membrane isolating the DNA. Ribosomes, represented by these little polka dots. Cytoplasm is clear in this model, and it's the jelly-like material filling the cell. A plasma membrane. A cell wall surrounds the plasma membrane and is very rigid. It holds everything into a specific shape. A capsule. Many prokaryotic cells have a capsule that's this outermost layer, and it's a gel-like coating outside of the cell wall. Pili are tiny hair-like structures, also known as fimbriae, and they act like Velcro to allow the bacteria to stick to anything. There are also needle-like pili used to exchange material between different prokaryotic cells. Flagellum are whip-like structures used in movement, and there can be anywhere from 1 to 5 flagella sticking out at one end that moves to propel the cells through their watery environment. The second main type of cell is eukaryotic cells, and we will review them in later videos. Thank you for watching this overview.